Warm welcome to lecture 4th of the cell biology. Myself Dr. Sunil Patil from LUH College Panchoti, Nashik. In this lecture we are going to see the plasma membrane. In the plasma membrane, unit membrane concept, the models lipid membrane, protein-lipid, that is Danielli-Davson model, and fluid mosaic model. The plasma membrane, also called as the cytoplasmic membrane, cell membrane, the biomembrane. It is commonly called as the plasma membrane. The plasma membrane, it is thin, cosy fluid structure present both extracellularly and intracellularly. Extracellularly it is present around the protoplast, and intracellularly it is present around the organelles. Around the cell, same membrane is there. Around the organelle, same structured membrane is there, called as the plasma membrane or cytoplasmic membrane. Thickness of plasma membrane is about 75 angstrom. The plasma membrane of eukaryotic cell is a phospholipid bilayer containing proteins like prokaryotic membrane. Movement of material across the plasma membrane occurs when passive and active processes, as in prokaryotics. You can see in the figures the structure of plasma membrane, phospholipid bilayer. Some characteristic features of plasma membrane: the cell membrane or plasma membrane, it is biological membrane that separate interior of the cell from the outside environment. The cell membrane is selectively permeable to the ions, organic molecules, and controls the movement of substances in and out of the cell. The basic function of the cell membrane is to protect the cell from its surrounding. It consists of phospholipid bilayer with embedded protein. Cell membrane are involved in variety of cellular processes such as cell adhesion, ion conductivity, and cell signaling. It also serves as the attachment surface for several extracellular structures, including the cell wall and the intracellular cytoskeleton. The membrane is selectively permeable and able to regulate what enter and exit the cell, thus facilitating the transport of material needed for the survival. So this is the structure of plasma membrane. We are going to see the structure in detail now. This is the cell. This is the cell membrane, phospholipid bilayer, and this is structure of phospholipids. The first in our syllabus, that is unit membrane model, that is protein-lipid bilayer and protein, means protein from both side, and middle one is the lipid bilayer. So 4 layers are there, 2 layers of lipid and covered by the protein layers. Robertson, it is name of scientist, who given the unit membrane model, given by the J. David Robertson in 1959 after the biomembrane model, which was proposed by Daniel-Davson in 1935. This biomembrane model proposed by Robertson stated that the biomembrane are made up of 4 layers. Out of these 4 layers, 2 are protein layers and 2 are phospholipid layers. Phospholipid bilayers is 25 to 35 degree thick, and each of 2 protein layers is 20 to 25 degree angstrom thick, overall 65 to 85 angstrom. Robertson used the electron bombardment of membrane and observed 2 distinct zones. So these are 2 inner lipid bilayer and outer is the protein structures. So this is molecular structure of the unit membrane model. The Robertson unit membrane model, a unit membrane has thickness 75 degree, 2 or more layers made of this. Each of 2 electron dense surface layer is 20 degree thick, and it is formed of extent of B protein molecule. Outer protein layer consists of mucoprotein and inner protein layer consists of non-mucoprotein. Central electron transport layer is 35 angstrom thick, which is a biomolecular layer of phospholipid. Drawback of this model was doesn't explain permeability and elasticity of the plasma membrane. So this is again the same structure of the unit membrane. Lipid bilayer model. This model to explain the surface of plasma membrane was given by Orton, Gorion, and Grendel. In 1902, Orton observed that substances soluble in lipid could selectively pass through the membrane. On this basis, he stated that the plasma membrane is composed of thin layer of lipids. Coulter and Grendel 1926 stated that the plasma membrane is made up of double layer of lipid molecules. This model, Coulter and Grendel, could not explain the proper structure of plasma membrane, but they put the foundation of future model membrane. The only lipid. Then Danielli-Davson model, it is also called as the sandwich model of cell membrane. The Danielli-Davson model, it is a post-molecular model. It was model of the plasma membrane of cell proposed in 1935 by Davison and James Danielli. The model describes a phospholipid bilayer that lies between two layers of globular protein, and it is trilaminar and lipoproteinous. It was the first model that attempted to describe the position of protein within the lipid bilayer found in the membrane. Danielli-Davson proposed this 4-layer model. It is protein flanked bilayer structures. The model was also described a lipoprotein sandwich structure. The lipid layer was sandwiched between two protein layers, so it is called as the sandwich model also. Danielli-Davson model predominantly until Singer-Nicholson model comes, that is fluid mosaic model. The fluid mosaic model expanded on the Danielli-Davson model, expanded form of Danielli-Davson model. It was the base of fluid mosaic model previously proposed by the flanking protein layers around the lipid bilayer, sandwiching them. Key features of the Danielli-Davson model, it is proposed model called as the sandwich model for membrane structure, in which a lipid bilayer was coated its either side with the hydrated protein, that is globular protein. Plasma membrane might be composed of two lipid protein bilayers, one facing the interior of the cell and other facing to external environment. The association between the surface protein and the biomolecular lipid bilayer, it is maintained by electrostatic forces. Such a membrane would exhibit selectively permeability, being capable of distinguishing between the molecules. They predict the lipid bilayer thickness, and each of the protein layer is about 1 nanometer thick, a total giving about the total thickness, it is 8 nanometer. Protein coat the outer surface do not permit the lipid bilayer structure. Then fluid mosaic model, which is the well accepted, it is most accepted model cell membrane. It was proposed by Singer and Nicholson in 1972. Fluid mosaic model is made up of phospholipid bilayer and protein. Proteins are like iceberg in the sea of lipid. Protein can change their position. Some proteins are intrinsic, means that occur at the different depths of the membrane. They span of entire thickness of the membrane, so they are called as the transmembrane proteins, which are present from outside to inner side of, they are transmembrane, entirely passing through the membrane, the transmembrane protein. They form channels for the passing of water. Extrinsic or peripheral, the second type of protein, extrinsic protein, they are found on two surfaces of the membrane. So this, some proteins, they are intrinsic protein, some are extrinsic proteins. Intrinsic protein and extrinsic protein shown in the figure. According to Singer and Nicholson, the membrane is calcified, in which lipid forms the viscous two-dimensional solvent where protein are inserted to integrated more or less deeply. The membrane is selectively permeable during passive transport. Many molecules move across the membrane without spending energy. Some solute move by simple diffusion along the concentration gradient from the higher to lower concentration. Neutral solutes may move across the membrane by the process of simple diffusion. This is called as the passive. Water also move by the osmosis, that is passive transport. Phospholipids is an amphipathetic molecule. This is structure of a phospholipid, meaning it has both hydrophilic head and hydrophobic region, so called the amphipathetic. In this phospholipid bilayer, the proteins are seen to float just like the iceberg in the sea. Most of the plasma membrane contain lipid and proteins, as the major components of the carbohydrates are in small component. There are 5 major phospholipids found in the plasma membrane, that is phosphodiecholine, then phosphodiol ethanolamine, then phosphodieserine, then sphingomyelin and phosphodiol linositol. Cholesterol and sphingolipids clustered in the small semi-solid particles, highly organized structures called as the lipid rafts. So this is structure, these are the head region, these are the tail region. They are hydrophilic head and hydrophobic tails, hydrocarbon tails. So this is the structure of the plasma membrane. There are two classes of proteins, peripheral and integral. We have already seen peripheral proteins dissociated from the phospholipid bilayer very easily. They are attached to the phospholipid bilayer by means of protein-protein interaction. Example is the spectrin and the ankyrin and actin. Integral protein have one or more segmented embedded in the phospholipid bilayer. For removing integral proteins, it is necessary to break off or disrupt the phospholipid bilayer. Most of the integral proteins are transmembrane proteins, means they pass throughout it, which span the membrane on both sides. That is glycophorin, band 3, purine are the examples. Then we are going to see the other structures. Carbohydrate portion comprises of the glycolipid and glycoprotein. Glyco means sugars. Sugars with lipid, sugars with protein, means carbohydrate with lipid, carbohydrate with proteins. The glycolipids are exposed on the outer surface of the plasma membrane. The carbohydrate coat present on the cell surface is called the glycocalyx. It is formed by oligosaccharide of the glycolipid and transmembrane glycoproteins. Structure and function features of the fluid mosaic model. Mosaic in nature, fluidity membrane. The mosaic nature means the membrane is due to the unique composition and has been proved that result freeze etching technique. That is, proteins are shown in the plane of cleavage of lipid bilayer, and fluidity is contributed by unique motional properties of the lipid bilayer, such as flexion, lateral diffusion, flip-flop motion and rotation. So these are the structure motional properties of lipid. You can see the flip-flop motion when diffusion takes place in the axial rotation. The integral proteins bind asymmetrically to the lipid bilayer. All integral membrane proteins have specific orientation. All the molecules of any particular integral protein, such as glycophorin, they move in the same direction, unidirectional. In contrast to the phospholipid, protein have never been seen in flip-flop movement, that is across the membrane, because such movements would be unfavorable for the energy consideration. Semi-permeability of the plasma membrane. The plasma membrane regulates entry and exit of material. It permits passage of certain substances and restricts other some substances. This property of membrane is called the selective permeability. This property depends on the several factors that are related to the structure of the membrane. The substances that are dissolved in lipid pass easily through the lipid bilayer plasma membrane. A very few small uncharged molecule can pass through the lipid bilayer. The lipid bilayer is impermeable to all change molecules or ions. Only a few change molecules can pass membrane via the pores and channels or carried by the transporter. Protein in the membrane help many substances to across the membrane. So this is about the plasma membrane. Thank you very much. In next lecture we are going to see the topic from the same chapter, that is membrane receptors. See you again.